All right, so now that you kind of have an idea of the stability of DNA, as long as it's preserved correctly, let's get into how DNA was discovered. And that really happened with three foundational experiments. So, this first experiment is shown by table 10.3 in your text, even though it says 10.2 here, was the discovery of the transforming principle. And this was done by Fred Griffiths in 1928. And he was an English physician who was really specialized in pneumonia and specifically Streptococcus pneumonia. So, Dr. Griffiths had actually succeeded in isolating several varieties or strains of pneumonia, type 1, 2, 3, and 4. And the disease causing form of the strain he found was actually surrounded by a polysaccharide coat, and that surrounding polysaccharide coat really made the bacterial colony appear very smooth when it was cultured. So, what he did was he actually found that the virulent form occasionally mutated into a non-virulent or non-infectious form. And that non-infectious form tended to lack that polysaccharide coating, so the edge of the cell looked very, very rough. So what Griffiths observed was that there was a small amount of living type 3S bacteria. Okay, and so he isolated it and injected it into the mouse, and that caused the mouse to develop pneumonia and die later. And when he was posting the mouse after it had died, he found large amounts of this type 3S bacteria in their blood. Okay, so the next step that Griffiths took was he actually injected a type 2 resistant bacteria or a type 2R bacteria, and the mouse didn't come down with pneumonia and he didn't die. So then Griffiths had also known from previous research and work with colleagues that if you boiled and killed all the bacteria, or if you boiled a sample or a set of bacteria that was infectious, you could actually kill it. So what he did was he heated up the type 3S that he originally had put in that first mouse that died. He heated it up, injected it into the mouse, and the mouse lived. So the theory was substantiated: as long as you heat it up, then it is going to kill the bacteria. So the last step, the last major question that Griffiths wanted to address was what would happen if you took the type 3S infectious bacteria and mixed it with the non-virulent bacteria. And he was fully expecting the mouse to live, but what ended up happening is he injected it into the mouse. Over a couple of days, the mouse slowly developed pneumonia, and on day 5, he died. And so when he was posting the mouse and collecting tissues of that mouse that died, that fourth one that died, he was able to collect in the blood, he observed the live type 3 bacteria that was the originally infectious one. So, what ended up, and this was not what he theorized would happen at all. So, what he ended up identifying was that type 3 bacteria that was infectious had the ability to transform itself and had the ability to be inherited to the next generation of the cell. So Griffiths had a couple of theories about how this was actually happening. So his second major theory that he looked at was that the live type 2 resistant bacteria had actually mutated to the virulent S form. And that virulent S form actually was the one that caused the pneumonia and allowed the mouse to die. And so this really was led to the identification that there's some sort of material that is in the polysaccharide coat of the dead bacteria cell that is responsible. So he started calling that the transforming principle. So the next step was to identify what the heck this transforming principle was. And so about 10 years later, Avery, Cullen, and McLeod, and then in addition to McCarty, started working on isolating and transforming that substance. So what they ended up doing was taking that type 3S bacteria that Griffiths had worked with, and they isolated it and then treated it with enzymes to destroy the proteins, the DNA, or the RNA. And keep in mind, they didn't know what RNA was, they didn't know what DNA was at this point, so they were just treating it with things that they knew would kill other things. So what they ended up finding was that if you treat the sample with RNase or protease, so allegedly killing the RNA and the proteins, what you find is that the cultures actually will still or will contain transformed virulent or infectious bacteria. So just because you added those substances means that it didn't kill that infectious bacteria at all. It was still very much alive and well. However, when they added DNase to it and killed the substance that killed the DNA, all that they got was the type 2R bacteria, the resistant type. So they knew that that third substance that they were treating this with was the thing that was actually killing the and not allowing that generation to be produced inside the animal. So the next step was to actually try to figure out how this was happening. So Hershey and Chase, what they ended up doing was that they wanted to make sure or to confirm that this DNA material was actually the genetic material. And so what they did is they took a T2 bacteriophage, which is a virus that infects E. coli. And the phage actually reproduces by attaching to the outer wall of the bacterial cell and then injecting its DNA into the cell, just like an alien movie. Okay, and once it's in the cell, it replicates and directs the cell to actually start synthesizing phage proteins. So the phage DNA becomes encapsulated within the phage proteins, producing progeny that eventually, if we look at steps 4 and step 5 down here, are going to lyse and break open the cell and escape into the system. So that's a little bit scary as well. So Hershey and Chase were able to grow up this batch of E. coli in a certain medium, and then actually were able to label the DNA of the phage with a certain marker called 32P. And in the second batch that they grew up, they labeled that with a different marker called 35S. And then they infected the bacteria with a T2 phage so that all the protein phages would have proteins labeled with the 35S. So what they ended up finding in this study was that after the bacterial cells were lysed and new progeny phages emerged, many of the phages actually emitted radioactivity because of those markers that they had attached to them. And so that really demonstrated that DNA from the infecting phages had actually been passed on through the progeny. And so that result again confirms that it's DNA, not protein, that is actually the genetic material jumping from one generation to the next. All right, so I wanted to just give you guys a little bit of background about the studies that were put in place to identify this DNA chemical. And now in the next section, we're actually going to jump into the structure of DNA.